All right, welcome to Chapter 3 of Human Anatomy and Physiology. This chapter is going to focus on cells. This should be another review from our basic biology understanding sophomore year. So you also be giving an online assessment with this as well. We will do a very brief discussion of Chapters 2, 3, and 4 in class. But to save time and hit other topics in advanced human anatomy and physiology, we're just going to use these as a review. So, as an introduction, we know that the cell is the basic unit of structure and function in living things, and cells vary in their shape, size, and arrangement. But all cells have similar components, each with a particular function. As cells divide, they differentiate or become specialized. So when we think of cells, cells are that basic unit of structure and function in the human body. And overall, there are about 75 trillion cells. And when we measure the cells, some of them are very microscopic. The cell size, typically when we measure them, is in micrometers. And when you look into the microscope and the ocular lens, you could see a little ruler that would be a micrometer. So your little micrometer will be able to use for measurement of things under the microscope. So differentiation itself is when cells differentiate from a common cell into their specialized cell type. And then as a result of differentiation, you get cells that are going to vary in size, shape, so on and so forth. And basically, they're going to vary their structure, so their structure is going to meet their function. And that's what anatomy and physiology is all about, structure and function. How does the structure of something meet its function or physiology? Anatomy and physiology. So when you look at a composite cell, also called a typical cell, the major parts include the nucleus, cytoplasm, and the cell membrane. So a typical animal cell, as illustrated here on the slide, has those three parts: the cell, or the plasma membrane, cell membrane or plasma membrane, which is that outer boundary of the cell. And then on the inside, you have the cytoplasm. And remember that the cytoplasm is going to be the area that holds all those cellular organelles and performs specific functions of the cell. The cytoplasm is considered to be the area between the plasma membrane and the nuclear region. So that would all be the cytoplasm. And then the substance that those specialized structures or organelles are suspended in is called the cytosol. And then, of course, in the center of the cell there, you have illustrated the nuclear region, or the nucleus, and that will be the control center of the cell. And in there, remember that the nucleus has the nuclear envelope that has pores. You also have the nucleolus inside there. And then, of course, what controls cell function would be the DNA. And in the undividing cell, that DNA is in the form of chromatin. So looking at the cell membrane, its general characteristics is that it forms the outermost boundary of the cell and allows the cell to receive and respond to incoming messages. It has a single transduction, and that will be discussed later in Chapter 13. Membrane structure is considered that fluid mosaic model. Now remember, a fluid is anything that has the ability to flow. So the cell membrane, which is composed of those phospholipids, that isn't static around the cell or stationary. It flows around the cell. It moves, and those phospholipids are changing position with one another. And then mosaic, when you think of mosaic art includes many different components of that art. So you could look at a mosaic art piece or picture, and it can include pieces of glass, rocks or stones, tile, and all those things will be brought together to create a very aesthetic picture that you could hang somewhere. Well, the cell membrane, although the major component is the phospholipids, you also have embedded in there fats, proteins, glycoproteins, so on and so forth. So that's where the mosaic comes from. The cell membrane is composed of a double layer of those phospholipid molecules with many proteins that are going to be dispersed in it. Peripheral proteins, integral proteins, channel proteins, so on and so forth. The surfaces of the membrane are hydrophilic due to the polar phosphate heads of that phospholipid. And then you have an internal portion of the membrane that is hydrophobic due to the non-polar fatty acid tails. So remember that a hydrophilic, those would mean that the heads are water soluble or like water, and then hydrophobic, those would be lipid soluble because they're fatty, so they'd be soluble in lipids or a fatty substance, but they'd be insoluble in water because they dislike water. They have that phobia. The membrane proteins also have the hydrophilic and hydrophobic properties, and there are basically 5 types of membrane proteins. The integral proteins, they are firmly inserted into and extend across that entire lipid bilayer, and they're going to serve as either channels or pores in the cell membrane, or transport proteins, so those would be called carrier proteins, or we also have integral proteins that are called receptor proteins, and that's going to serve as an aid in recognition sites for those proteins. And then of course you also have enzymes. And then they're in the clinical application, it talks about faulty and ion channels and drugs and how they play a role in the effect of ion channels. In addition to that, you have receptor proteins, enzymes, you have cell adhesion molecules that are going to guide in cellular movements, and you also have those cell surface proteins. So here is a good example of what the cell membrane looks like. So you can see all those glycolipids, carbohydrate glycoproteins, fibrous proteins, the phospholipids themselves, how they're a bilayer, globular proteins, cholesterol. Cholesterol is important in the cell. It helps play a role in cell adhesion. So you get a good idea of what that cell membrane looks like there. Okay, moving forward. Cell adhesion molecules, or CAMs. Cell adhesion molecules are help guide the cells on the move. You have selectin, which allows white blood cells to anchor. You have integrin, which guides white blood cells through capillary walls, and important for growth in embryonic tissue, and they're important for growth of the nerve cells. The cytoplasm is a network of membranes and organelles suspended within the cytosol. The cytoplasm also has that cytoskeleton, which forms the supportive framework, and it's made up of protein rods and tubules. So the cytoplasm, often the cytoplasm and cytosol are used interchangeably, but the cytosol itself is that jelly-like fluid, which is about 70% water, that holds the cell organelles and occupies the space between the nucleus and the cell membrane. It contains an abundance of proteins and protein rods and tubules that help give the form of the cell and support its framework. That would be the cytoskeleton coming into play there. And then, of course, within the cytoplasm and embedded in the cytosol, suspended in the cytosol with the help of the cytoskeleton, you would have organelles, or little housing compartments, where their structure is going to help meet their function of the cell. So each of these organelles is going to have a specific job. And in there we have several. The first one you could see there is the endoplasmic reticulum. And the endoplasmic reticulum is that inner membrane network. So endoplasmic reticulum is a network, and endoplasmic means inside the membrane. It's kind of like we have an endoskeleton. Endoskeleton means inside our body. insects have that exoskeleton outside. So you have this inner membrane network of sacs, canals, and vesicles. And basically there are two types of endoplasmic reticulum, or ER, within the cell. You have the rough endoplasmic reticulum, or rough ER, and then you have the smooth endoplasmic reticulum, called the smooth ER. Basically, the difference between the two, the rough ER is studded with ribosomes, and it's going to play an important role in the functioning of protein synthesis. Smooth ER is smooth because it lacks ribosomes, and its important role is the synthesis of lipids and cholesterol synthesis. It also helps store calcium ions, and we'll talk about calcium ions when we get into muscle contraction. And it's also abundant in liver cells, and there's where you have hepatocytes. So the endoplasmic reticulum, again, is that connected membrane-bound sacs, canals, and vesicles. And basically, it's that interconnected parallel membranes, or mazes, that is continuous with the nuclear membrane. So it extends right off of the nucleus there. Vesicles are going to be transport sacs, and they are going to be made by both the endoplasmic reticulum and the Golgi apparatus, and we'll talk about that a little later. You also have ribosomes, and ribosomes can be both free-floating or connected to the ER, which would make it the rough ER. They provide structural support and enzyme activities, and they are going to help form proteins. This would be the site of protein synthesis. So in general, they're small granules that are dispersed throughout the cytoplasm and on the membranes of some of the ER. They are composed of RNA and protein. So you have that rRNA functions in protein synthesis, and these little structures themselves are not bound by a membrane. The Golgi apparatus complex, the Golgi apparatus is actually flattened membranous sacs called cisternae, and those are going to be arranged in stacks, kind of like stacks of pancakes, and associated with many vesicles or membrane-bound sacs that would contain proteins. The function of the Golgi apparatus, sometimes referred to as dictyosomes, is going to be in the modification, packaging, and transport of proteins that are going to be made by the ribosomes. The vesicles will pinch off as secretory vessels, which are going to be transported out of the cell. And of course, that entire process, that's a form of exocytosis there. So you can see what those would look like in the diagrams on your slide. Then you have some energy-related organelles. One of those energy-related organelles that we studied in biology were mitochondria. Mitochondria, or mitochondrion, which is plural, these are kidney-shaped organelles whose inner membranes are folded into shelf-like portions called the cristae. So remember, here you have a double membrane-bound organelle, has an outer membrane, intermembrane space, and an inner membrane, and those inner membranes are folded to form cristae, and this would be the site of cell respiration. And because it's the site of cell respiration, we call it the powerhouse of our cell, because this is where energy is going to be released from glucose, and we're going to form ATP. Mitochondria does have its own DNA, mitochondrial DNA, and you can read about that a little more in your textbook. And there are also some related mitochondrial diseases that you could read about in your clinical application of the textbook. So here you see the interaction of organelles. So if you look here, you have lipids are being synthesized in the smooth endoplasmic reticulum. Milk protein genes are transcribed into messenger RNA. The messenger RNA exits through the nuclear pores. Most proteins are synthesized and ribosomes associate with membranes of the rough ER, using the amino acids in the cytosol. The sugars are synthesized in the smoothie and the Golgi apparatus and may be attached to the protein or secreted in a vesicle. And then the proteins are secreted from the vesicles that butt off of the Golgi apparatus, and then fat droplets will pick up a layer of lipid from the cell membrane as they exit the cell. And that would be exocytosis occurring there. Lysosomes, these are spherical membranous sacs containing digestive enzymes or proteins. Sometimes people call these suicide sacs because they're going to destroy anything the cell no longer wants or needs. I kind of don't like suicide sacs. I like to call them the garbage disposal of the cells. Basically, it is a process of autolysis, and that is the process by which worn cell parts are going to be digested by autophagy. And you could see, again, some more clinical applications as far as organelle-related diseases in your textbook with these guys. Peroxisomes are membranous sacs that contain oxidase enzymes. If you remember, peroxisomes are going to play a role in detoxification of harmful or toxic substances. Peroxisomes would detoxify things such as alcohol, formaldehyde, oxygen-free radicals, which are bad to the body. They also break up hydrogen peroxide into both water and oxygen gas. And you can remember that when we used catalase, the catalase enzyme in our lab, and we used the cow's liver to break down hydrogen peroxide into its individual components. Centrosomes, these are a pair of microtubules located near the nucleus, and they aid in the movement of chromosomes during mitosis. So that would be related to cell division. Centrosomes are used to produce the cilia and the flagella, and they are basically two rod-like centrioles. If you look at cilia themselves, cilia is plural, the cilium singular. Cilium themselves are short hair-like cellular extensions, much like eyelashes, that help move substances through passageways, and they are located in the lining of a trachea and the fallopian tubes. The flagella and the flagellum, singular, that's a tail-like projection, only one flagellum per cell in humans, and that is going to aid in cell locomotion because it's going to provide motility to sperm cells. Microtubules and microfilaments in cells. Basically, microtubulants and microfilaments are protein structures, and you have, of those protein structures, you have the microtubules, microfilaments, and you also have intermediate filaments. And these are going to help form muscles and bones of the cell, and they're going to allow for intracellular transport and movement. Also in the cell, you have, if we come back to here, I don't see it on a picture. Oh, nope, not pictured. All right. Also in the cell, you have things called microvilli, and the microvilli are going to be small finger-like extensions of the external surface of the cell membrane, and that's going to be used to increase the surface area. All right, the nucleus. The cell nucleus is the central core, control center, or brain of the cell. It's the largest organelle of the cell. It's filled with nucleoplasm, and it contains three distinct regions. You have the nuclear envelope, the nucleolus, and the chromatin. The nuclear envelope is a double membrane bound it's a double membrane bound barrier that's going to help separate the internal nucleus from the external nucleus. So it's going to keep the components of the nucleus inside from the rest of the cytoplasm of the cell. At various points, these two membranes will fuse uh, with nuclear pores. So you have those nuclear pores that helps allow things to transport into and out of the nucleus. Like the cell membrane, the nuclear membrane is selectively permeable, and the pores are going to serve as sites where the messenger RNA is going to pass out of the nucleus during protein synthesis, and how those ribosomes exit the nucleus. The nucleolus itself is those dense spherical bodies that lie within the nucleus. You could have nucleoli, which would be plural, and those are going to be composed of RNA and proteins, and it's going to be in the function, as far as function of the nucleolus, it's going to serve in the production of ribosomes. So those ribosomes are also going to use those nucleopores in the nuclear envelope to get out into the cell, where they could be free floating or attached to the rough endoplasmic reticulum. And then lastly, you have chromatin, and chromatin is going to be loosely coiled fibers of DNA and histone proteins that is going to be present in the nucleus. And if you remember DNA and those histone proteins, when we talked about DNA packing in biology, you had the chromatin wrapped around those histone proteins, and then you had a cluster of histone proteins and chromatin that would form the nucleosome, and then you could get the supercoiling. So that's how we get all that DNA in our cell there, and eventually that would become, can become a visible chromosome there. So when the fibers of chromatin will be condensed into tightly coiled chromosomes, the cell then would be preparing for division. Okay, so that is a lot of what we did with structure and function inside. So as far as the structure of the cell, its individual components, and their function, the other part of biology we looked at was movement into and out of the cell. So this would be related to membrane transport. And when we looked at membrane transport, basically we said it was the passage or movement of substances through the cell membrane, and it may be physical, such as passive transport, where it requires no energy expenditure in the form of ATP, or it could be physiological, which would be active transport, and that's going to require the expenditure of ATP. So in physical or passive transport, the substances are going to move from where they are in high concentration to where they are in low concentration. And passive transport processes include simple diffusion, facilitated diffusion, osmosis, and filtration. In physiological or active transport mechanisms, the substances are going to move from where they are in low concentration to where they are in high concentration at the expenditure of cellular energy that we call ATP. It's an active process that includes both these mechanisms of active transport, endocytosis, exocytosis, and then transcytosis. So in physical passive transport, it requires no energy. In simple diffusion, illustrated here, you can see molecules or ions spread spontaneously down a concentration gradient until a state of equilibrium is produced. And examples of this process include a sugar cube dissolving in water, a drop of dye diffusing in water, and odor diffusing throughout the room. It also includes the diffusion of oxygen and carbon dioxide gas through the cell membranes. Simple diffusion plays a significant role in human metabolism, and it is key to cellular respiration with the exchange of gases across the membrane. In facilitated diffusion is a special case of diffusion where you're going to have substances move with the concentration gradient from high to low, so it's still a passive transport, but it requires a special carrier protein molecule within the cell membrane that is going to serve kind of as a shuttle bus to transport molecules into and out of the cell. It's significant because this process is by which most of the glucose in our cells is going to enter and leave. So glucose will enter and exit the cells via facilitated diffusion. So again, it's another important part of cell metabolism as far as cell respiration, bringing that glucose molecule in. And then you have a special type of passive transport called osmosis. And osmosis is the diffusion of water molecules through a selectively permeable membrane. And it's an attempt to dilute a particular solute. So remember, a solute is a substance that is going to be dissolved in water. Remember that water can pass through the membrane, but a solute cannot. So osmosis is significant in maintaining the osmotic pressure of our cells. And the osmotic pressure of our cells is at about 0.9%. The solute dissolved in the water of our cells, tissue fluid, and blood measure about 0.9% saline. So when the solutions are infused into our body or tissues, into our blood or tissues, the solute concentration of the solution must be equal to that of our cells and tissues. So it needs to be an isotonic condition, which would be 0.9% then. If you are not at that 0.9% or that isotonic state, then that's when you're going to have water either enter the cell or leave the cell. So if we lose water, the cell will shrink. If we gain water, the cell will swell up. And because animal cells lack that cell wall that plant cells have, we only have that cell membrane, that cell could burst or lyse itself. Osmosis is demonstrated nicely here in this picture using erythrocytes or red blood cells. So you can see that osmotic pressure is the ability of osmosis to generate enough pressure to move a volume of water. Osmotic pressure increases as the concentration of that non-permeable solute increases. And then here you can see the three osmotic conditions of varying tonicity. And tonicity is when they are being placed in those solutions, the pressure that's going to be exerted on that cell membrane. So they would be isotonic. Iso means same, so you have the same osmotic pressure on both sides of the cell membrane, so you're not going to have no net gain or loss of water there. You're in an isotonic state. In a hypertonic state, you have higher osmotic pressure, so you're going to have water loss, and the cells placed in a hypertonic solution are going to shrivel up or shrink. And then in a hypotonic state, that is when you have lower osmotic pressure, water gain, and the cells are going to be placed in a hypotonic solution, will be gaining water, therefore gaining mass, and that's when they could burst or lyse themselves. Lysis is when they burst. You also have filtration. Filtration is water and solutes which are forced through a body membrane by the hydrostatic pressure of blood, which we would call blood pressure then. It's a concentration gradient is from high to low. The solutes include both glucose, gases, ions, hormones, and vitamins. So an example of this is excretion. Blood being filtered through the capillaries is going of the kidneys is going to be used to help remove those waste. So smaller molecules are going to be forced through porous membranes. Hydrostatic pressure is important in the body, and the molecules leaving those blood capillaries. Active transport. Remember that active transport, we do have an expenditure of energy in the form of ATP here, because the molecules or ions are moving from areas in which they are in low concentration, and they're going to move to an area of higher concentration. Substances include many ions, amino acids, and monosaccharides. Glucose, fructose, galactose are examples of monosaccharides there. And also here we hear of the sodium-potassium pump, which requires ATP, which maintains, when we look at the sodium-potassium pump, that is going to play a huge role in the nervous system when we talk about resting membrane potential of the nervous system. So active transport, the sodium-potassium pump, it's an active transport mechanism. It's going to create a balance by pumping 3 sodium ions out of the cell and bringing 2 potassium ions into the cell. So it is a 3-to-2 ratio there. Endocytosis. Basically what's happening here is you're bringing something into the cell, cytocell, endo in. So endocytosis, molecules or particles that are too large to enter the cell via means of passive transport or active transport above are brought into the cell within a vesicle. So it's formed from a section of the cell membrane. And there are 3 types of endocytosis. You have pinocytosis, and pinocytosis is cellular drinking, and the cell is going to bring in liquid droplets which may contain dissolved substances. You also have phagocytosis, and that's cellular eating, and that's when the cell is going to engulf and bring in solid particles. Phagocytosis or macrophages are very important scavenger white blood cells in humans. They will also bring in foreign particles like bacteria, etc., that are going to then fuse with the lysosome, and their cytoplasm of the lysosome is going to help digest those foreign particles that are in the cell. And lastly, you have receptor-mediated endocytosis. And receptor-mediated endocytosis requires the substance to bind to a membrane-bound receptor. We could talk about, if in your book, we will look at this a little bit, on page 106, it looks at LDL receptors and then some drug application there. So here you see a picture of endocytosis. Up here you have the different types. This would be receptor-mediated endocytosis, so on and so forth. Exocytosis. In exocytosis, it's physiological transport. In this process by which the cell is going to transport secretory proteins out of the cell. It also allows cells to get rid of debris by dumping it to the outside. So that is into that extracellular fluid. Basically, as far as our studies in anatomy and physiology, it's going to be releasing of a lot of neurotransmitters, again, in the nervous system. And we'll talk about that as you release those neurotransmitters across the synaptic cleft. And then you have transcytosis. This is something that we did not talk about back in 10th grade biology. But transcytosis is a combination of endo and exocytosis. And basically the particles are going to travel across the cell from apical to basal surfaces. And it enables the immune system to monitor pathogens in the small intestine, preventing food poisoning. It's also the way that the HIV virus is going to pass through our mucous membranes. And you can see that illustrated in the diagram here. So there you have a picture, which be a lining of the anus or the vagina. Ultimately, that would be epithelial cells. And there you could see HIV-infected white blood cells. And the HIV would be these little green particles, and how they would pass across receptor-mediated endocytosis into the cells from external to internal in the anal or vaginal canal. Very brief, going over the cell cycle. The cell cycle itself, it is a series of changes in a cell that undergo from time to time as it forms until the time it divides. The stages itself include interphase, and then you have the M phase, which would include both mitosis and cytokinesis. In interphase, the cell is part of where it will have growth and DNA replication. It's not part of mitosis. It's represented by the majority of the cell's life and includes cell growth and duplication of DNA during its S phase prior to mitosis, that first stage of mitosis, which would be prophase. Interphase is divided into 3 individual stages called G1, S, and G2. In the G1 phase, you have rapid growth and replication of centrioles. In the S phase, you have growth of DNA replication. And in the G2 phase, you have growth of final preparation for cell division. Cell division itself is the mitotic or M phase, and it's going to be in this stage where the cell is going to begin to divide into 2 new cells called daughter cells. And basically this process includes both mitosis, which is division of the nuclear parts of the cell, and then you also have cytokinesis, which is division of the cytoplasm, so everything else. Mitosis itself is done in 4 phases. In prophase, you have distinct pairs of chromosomes that become apparent as they are tightly coiled around DNA, and tightly coiled DNA occurs around histone proteins. The chromosome pairs are composed of identical sister chromatids that are held together by their centromere. The centriole pairs are going to migrate to the opposite ends of the cell. Spindle fibers are going to form between them, and the nuclear envelope and nucleolus are going to begin to disintegrate. In metaphase, basic characteristic of that stage is the chromosomes end up lining up in an orderly fashion midway between the centrioles, and we call that the equatorial plate of the cell. And then the centromeres holding the chromosomes together attaches to the spindle fibers between the centrioles at their kinetochores. In anaphase, the centromere holding the chromosome pairs together is going to split or separate, and the individual chromosomes are going to migrate in opposite directions on the spindle fibers toward the poles of the cell. And now we start to have cytokinesis beginning as we move into that last stage called telophase. And in telophase, the chromosomes are going to complete their migration toward the centrioles. The nuclear envelope develops around each set of chromosomes. The nucleoli develop and reappear. The spindle fibers are now going to disintegrate, and now you have cytokinesis beginning because you're going to start to have that cleavage furrow, which should be pretty much complete by this stage. So cytoplasm division, cytokinesis, that's at the end result. You'll have 2 new daughter cells, and it begins during anaphase when the cell membrane begins to constrict or pinch around the daughter cells, and then is completed at the end of telophase when the nuclei and cytoplasm of the 2 newly formed daughter cells are completely separated by that cleavage furrow. And here you can see, pause for a moment and check out the illustration of mitosis in the diagram here. Check it out in your textbook as well. So there you can see the what the chromosomes are doing. All right, control of cell division. Basically, cell division capacities vary greatly among cell types. Skin and blood cells are going to divide often and continually. Neuron cells divide a specific number of times, then will cease. You have chromosome tips, which we call telomeres, and those telomeres are going to shorten with each mitotic division, and it's going to provide a mitotic clock. The cells to divide to provide more favorable surface area. Remember that, maintaining that surface area to volume relationship or ratio. And you're going to have growth factors and hormones that are going to stimulate cell division. Hormones will stimulate mitosis of smooth muscle cells in the uterus. Epidermal growth factor stimulates growth of new skin. You have contact inhibition or density dependent inhibition We did take a look at that in our biology unit when we were talking about cell division. And then you have tumors that are a consequence of loss of cell division or cell control. And such tumors can be both benign and malignant. We also looked at this when we looked at mitotic cell division in our biology unit, as it fell under our cancer biology. So let's look at this a little bit more. Tumors, basically, abnormal cell division is cancer. And when the cell division occurs with no control, basically you get a tumor or growth or neoplasm results. And you could have two types, benign or malignant tumors. Malignant tumor is a cancerous growth. A non-cancerous tumor is a benign tumor. So malignant tumors may spread throughout the body via metastasis to other tissues by direct invasion through the bloodstream or lymphatic tissue. Basically, we know that in cancer biology, oncology is the study of tumors, and an oncologist is a physician who is going to treat patients with tumors. Basically, oncogenes, so the two main types of genes that cause cancers are oncogenes, which are going to activate other genes that increase cell division, and tumor suppressor genes, which normally regulate mitosis, but if inactivated, they are unable to regulate mitosis, and then the cells become immortal. So they just keep dividing, and dividing. Stem and progenitor cells allow for continued growth and renewal of the cells. A stem cell divides by mitosis to yield another stem cell and a partially differentiated progenitor cell. So basically, a progenitor cell is going to be a cell that is committed to a specific cell line. So it's going to differentiate, and it can differentiate into one of the four types of cells. It can become an epithelial cell, it could become a connective tissue cell, it could become a muscle cell, or it could become a nervous cell. And of stem cells themselves, basically you have those cells that are totipotent cells, and they can become any type of cell. You also have cells that are pluripotent, and pluripotent cells can become many cell types, but not all. So of the two, totipotent stem cells can give rise to any cell type in the body, and those are the ones, often when you hear about embryonic stem cell, that are most valuable. But when you look at non-embryonic stem cell research, a lot of those cells are pluripotent. And pluripotent, basically those cells would only give rise to certain cell types. So you're limited there. So progenitor cells are committed to the cell and can divide to become any of a restricted number of cells because they're pluripotent, and this all refers to cell differentiation. And basically cell differentiation or cell specialization occurs due to gene activation, and certain genes are going to cause those cells to become what they need to be. On the opposite side of generating cells and specializing cells, you also have the process of cell apoptosis or cell death. And apoptosis, which is programmed cell death, is a fast, orderly, contained destruction of cells that packages the cellular remnants into membrane-enclosed pieces that are going to be removed from the cell. It is a normal part of development. It sculpts organs from tissues that naturally overgrow, and it's important in fetal development as well. It is protective, so peeling of skin after a sunburn to prevent possible cancer. That is all part of apoptosis. And it is a continuous and stepwise process, much like mitosis is. So basically, what you have there is you get a death receptor on a doomed cell, or the cell's membrane is going to receive that signal to cause the cell to die. And then you're going to have an enzyme come in called capsaicin. And that enzyme is going to come in and it's going to start to destroy various cell components. And then the dying cell shape becomes round as it is cut off from other cells, and the cell membrane is going to undulate, forming bulges or blebs. And then the nucleus will eventually burst, the mitochondria will begin to decompose, and the cell shatters within 1 hour of the capacious release. So that enzyme there. And basically inflammation is prevented because the fragments are membrane encapsulated there, so you don't get any inflammation. Necrosis is a disorder form of cell death associated with inflammation and injury. So overall, this is our lecture on chapter 3, which is the cell chapter of our anatomy and physiology unit. So we looked at and explained how cells differ from one another. We looked at a composite cell, and we described the general characteristics of a composite cell, explained how the components of a cell's membrane provide its functions. We described each type, each kind of cytoplasmic organelle, and explained its function. We also described the cell nucleus and its parts. Then we moved on to talk about movement into and out of the cell and explained how those substances move. We looked at the cell cycle. We described the cell cycle and explained how the cell is going to divide into 2 new daughter cells. We looked at the control of cell division and described several controls on why cells divide. We looked at stem and progenitor cells and looked at how stem cells and progenitor cells can be used to make new cells and also play a role in growth and repair of tissues. And cell differentiation, we looked at how those cells are going to differentiate based on their cell types through genetic information, and how they'll have that different appearance and function. And then lastly, we concluded with cell death, and that was our discussion of apoptosis and the relationship between apoptosis and necrosis.